Morning, everyone. I think we might try and get started so that we can keep to time. I hope you all enjoyed conference dinner when you were out, and don't know if many people went onwards after they kicked out, but I didn't, and I'm still already a bit blurry. So this is the last, day, sort of half day of the conference, and I'm afraid someone has to do it, but someone has to have the plenary after the conference dinner, and I know Annette's lovely, so she would forgive me for doing it to her, so unfortunately it's Annette's turn to do the plenary after the late night out. Annette is a seabird biologist, but she didn't start out as such. She did start out in engineering, but then saw the light and joined our community instead, which is much, much better. But it does mean that she brings some really nice insights from her engineering background to seabird ecology. She completed her PhD in the University of Oxford and is now working for the Norwegian Institute for Nature Research. She is a National Geographic explorer and former Sky Ocean Rescue scholar. And her work focuses on using tracking technologies to record seabird movements while at sea. And one of the really interesting things she does is she tries to then link that to fitness consequences of populations, and that's what her talk's about this morning. So without too much further ado, I will give the floor to Annette.
Thank you, Mark. Thanks very much for inviting me to talk here, and thank you very much to all of you for turning up this morning. I have to say I'm impressed. I didn't expect that many people after the conference dinner, so thanks. So before I start talking about seabirds, I just want to acknowledge the very many people who have been involved in the studies I'm going to mention today, without whom none of this work would have been possible. So thanks very much to everyone, and quite a few are here today. So I study seabird movement, and for animals, movement is really essential in their lives. It's central to many behaviors, from migration, dispersal, to guarding a territory, finding food, or escaping a predator. And one of the questions I'm interested in is what drives animal movement, and what makes an animal goes where it goes, and what are the mechanisms behind this. And also what drives variation between individuals and groups of individuals in this movement. So basically, I'm interested to find out the mechanisms that are shaping a species' spatial ecology. But these different strategies may have implications for breeding success and survival of animals. So I'm also interested in the fitness consequences of these different strategies. And for seabirds, who are some of the world's greatest travelers, their movement, as you all know, is a really important part. Now, we can split movement in seabirds during two periods. Basically, during the breeding season, birds have to return to the colony to breed, and so they're central place foragers. And then during the non-breeding season, they go out at sea, and they're much more flexible. They don't have this constraint to come back. And during this talk, I'm going to focus about the latter period, so the non-breeding season after breeding. This is just a snapshot of the GLS data available for seabirds on the BirdLife seabird tracking database. And it's just to illustrate the wealth of data we have now on the migratory movements of seabirds. And you see some of the oceans, like the Atlantic, are completely filled up with points. There's some gaps elsewhere. That doesn't mean there's no seabirds there, obviously, but that we haven't yet tracked them. I'm going to talk about studies I've done in different species, but a lot of the studies I'm going to talk about today are done in puffins. For those of you who are not really familiar with puffins, just a quick introduction to the species. So they're long-lived seabirds, and actually the longevity record was broken just a couple of years ago by a puffin on Skoma that was recited, and it was 42 years old. And that's quite old. They nest in underground burrows and are monogamous. This is what they sound like. The nice soothing sound of a puffin. And this is what they look like. Very cute and charismatic bird. Here the behavior you see is called billing, and this is usually done between mates during the breeding season to reinforce the pair bond. They're not just cuddly, though, and actually anybody that has handled puffins will know that. They are quite feisty, and that beak can inflict quite serious injuries to the hand of researchers. So I started, and I still go there. I started studying puffins on Skoma Island in Wales over 10 years ago now. And my research on puffins on Skomer involves basically me waiting around all the time behind a scope, waiting for the puffins I've tagged to come back to their burrow. And the rest of the time, I'm just lying on the ground with my arm down a burrow trying to catch them. And in desperate cases, when I really can't catch them, I have to resort to hiding under a camouflage blanket, and then when the tag puffin returns to its nest, I have to sprint as fast as I can to get to the burrow before it leaves, because sometimes they only stay there for about 30 seconds. It's not always successful, but I have caught some that way. And so for the last 10 years, we've been putting geolocators on puffins, and actually this study started before I started on Skomer. It was started by Tim Guilford in 2007, so actually it's now been 15 years. And we've been putting geolocators on puffins to follow their non-breeding movements. And this was actually the aim. One of the key objectives of my PhD was to work out where the Skomer puffins were migrating. And the question of puffin migration has been fascinating researchers for a long time. People didn't really see big flocks of wintering puffins at sea, and so we didn't really know where they were going for a while. And there's even researchers in the 1940s that went on a plane and crossed the Atlantic over 100 times, counting the birds that they could see to try and find out where puffins migrated. And they crossed the Atlantic 111 times, and they counted 5 puffins. So this wasn't very successful. We got some insight from ringing recoveries, but really it's only more recently when geolocators became available that we started getting some insight into their migration. And the first puffins to be tracked were on the Isle of May by Mike Harris, Sarah Rennes, and Francis Daunt, and they found that the puffins on the Isle of May mainly winter in the North Sea. Then Mark Jessup did the same thing on Skellig Michaels, and then found that the puffins from Western Ireland are kind of using a large area in the Atlantic. And then I started my PhD and started doing the same on Skomer. This is just an animation of just a few tracks, all from the same year, of the migration of Skomer puffins. Obviously, ignore the parts where the birds are going over land. So there's quite a lot of interesting things here. The first one is that these birds are going really, really far. For anyone who's seen a puffin, they're not really cut for very long flights. They have really high wing loading, and flight is very expensive for them. So really, we didn't expect these birds to fly that far away, and especially some of the westwards movement are taking place in unfavorable wind conditions. So this was a big surprise. And actually, I still remember to this day when I first downloaded the first geolocator of that bird that was going all across the Atlantic to the Labrador Sea in just a couple of weeks. And I was sure I'd done something wrong with the data, and I'd plotted the wrong data, and I just checked again and again, and no, it was correct, and then we ended up getting more tracks. So it is quite impressive. The second thing in this animation is that it shows that all of these different puffins were going to completely different places, and that is quite an unusual migratory pattern for one population to completely disperse like this. Some were going to Iceland, some were going to the Labrador Sea, some were going just to the Irish Sea or the Mediterranean Sea. So this was really surprising, and we wanted to try and find out why this was the case. So although there was a lot of diversity between these individuals, I managed to classify these routes roughly in 4 groups. But bear in mind, there is some variation even within each group. The first group I called local, and basically you could also maybe call them even residents. So these are puffins who really didn't go very far. They stayed around the Celtic Sea, the Irish Sea, maybe the Channel and the Bay of Biscay, but really didn't go very far, and about 38% of them did that. Then there was a very small proportion of birds who did this for the first half of the winter, and then around December time, they then went to the Mediterranean Sea. And then there was quite a lot of birds, 44%, that had a much longer migration, and they all headed westwards towards the Atlantic. Now, how far west they were going kind of varied, but basically beyond that longitude that you see on this map, going across Iceland. And then they would do that in the first half, and then the second half of the winter, they'd start moving back towards Europe, and they would stay near the Bay of Biscay and the Portuguese coast. And finally, the longest distance migrants were doing this movement to the Atlantic first, and then they would go to the Mediterranean Sea. And that's thousands and thousands of kilometers. So we wanted to understand what was going on, and the first idea was, maybe they just go out randomly, whatever, and then this changes every year. So what we did is we tracked the same individuals multiple years. between 2 and 6 years. And we found that this wasn't random at all, because the same puffins kept going to the same place over and over again. And here you have one puffin that we tracked for 4 consecutive years. Obviously, there's a little bit of variation. Also, remember, this is geolocated data, so it's not hugely accurate. But basically, out of the 30 birds that we had tracked for multiple years in this study, there's only one that switched strategy. The 29 other ones just kept going to the same place. The one that switched basically had a local migration route for 2 years, and then it switched to an Atlantic route for 2 years, and then it switched back, which was quite interesting, and kind of suggesting a role of memory in this system. So we wanted to know if there was any benefit to having all this diversity of route, and maybe some strategies were better than others. And so we looked at the breeding success of these birds, because we had individual breeding success. So that's the breeding success after the migration. And we found that actually the birds that were going to the Mediterranean Sea were substantially and significantly more successful the next year than the birds that were sort of remaining local. And then these were more successful than those who were just going to the Atlantic but not the Med. And when we looked at their behavior using the wet-dry immersion data, we found that the amount of foraging that these birds were doing varied. Mostly they were just sitting on the water resting and not doing much flying. But those who were in the Mediterranean Sea spent more time foraging. And when we looked into it over like month by month, we saw, and this is the yellow curve on the right-hand graph here, that basically the birds in yellow, while the ones in the Mediterranean Sea, spent more time foraging significantly during the last part of the winter. And we know that during this time, around December time, there is usually a phytoplankton bloom in the Mediterranean Sea. And so it might just be that there's loads of food and this is a good place to go. So one of the ideas is that this could be that these birds are having a higher breeding success just because they are higher quality individuals and they end up on these migration routes. Or it could be that there's no difference in individual qualities, but it's just better to go to the Med because there's loads of food and you get in better condition before breeding. But in that case, why don't we see all the puffins doing this? Well, this study at this time and looked at 6 or 7 years of data, and that might just be a snapshot, actually, of the situation. So it might be that at that time, the Mediterranean strategy was better, but it could be that other times, those going to the Atlantic will do better. And actually now, since that study was done, we've now doubled the amount of data we had. We have 15 years. And so at the moment, I have a master's student who's now redoing this analysis and see if anything's changed over time. If you look at the migration strategies of monogamous birds that have long-time pairbond, there is evidence that if they synchronize returns to the colony before breeding, that helps them breeding earlier and being more successful. And because we found this huge diversity of routes in puffins, I was wondering how this affected pairs. And so we tracked some pairs of puffins and scoma to try and see if there was, for example, any similarities in their migration routes and how they worked out this synchrony problem. We found no real difference in the distributions of males and females, but what we found is that partners were following significantly more similar routes than two birds which were not paired. And you can see this on the graph in the right-hand side, which is on the y-axis nearest neighbor distance between pairs of tracks. And this is a measure that if it's high, it means two tracks are quite different from each other. If it's low, they're quite similar. And we can see that the tracks of partners were much more similar to each other than two random birds, but only during the first part of the season. And this is exemplified on the graph on the bottom left, which is just one pair. But basically, the birds head out in the same direction, but then at some point they kind of split. So the first idea was, are they migrating together? Because that would be quite a nice story, puffin pairs migrating together and spending the non-breeding season together. But they don't. We had to look, and there's obviously some too much distance between the two birds. So they are not together, but they head out in similar directions. But interestingly, we found that the pairs that had more similar routes then had a higher breeding success, laid earlier, and had a higher breeding success the year after. So there is some sort of advantage to doing this, even if they're not together. And it might be because if you migrate to a similar place, you are exposed to the same environmental cues that you might be using perhaps to time your return to the colony. And we did find indeed that pairs were more synchronized in their return to the colony than you would expect by chance. Another interesting thing we found in this study is that there is another predictor of lay date and breeding success in addition to the similarity of routes between pairs. And that is the amount of foraging that the female puffin does in the sort of last few months before breeding. Females that spend more time foraging lay earlier, and they have higher breeding success. And this is not visible at all in males. And one we think this might suggest is that the females might be already preparing for the egg formation. In procellariforms, usually, birds have like this pre-laying exodus period where the female goes away and forms the egg for a couple of weeks or sometimes longer and then returns to breed. But auks don't have this, and puffins don't. So sometimes when they come back to the colony, they have to lay an egg pretty quickly. And so it might be that these females are starting this egg formation and feeding for the egg formation really early on. So by this point, I had found out quite a little bit about the migration behavior of puffins on Skoma, and so I wanted to look at a slightly bigger scale. And luckily, during that time, many of my puffin researcher colleagues around the North Atlantic had also been deploying geolocators on their puffins. And so together, we put all our data together to obtain a sort of species-wide map of their distribution in the winter. And together we had collected data from 13 different colonies from all across the range. They breed in the Northeast Atlantic. And we covered all major key areas except perhaps Greenland. And in terms of sample size, we tracked nearly 300 puffins. And we obtained this rather complicated map here, which basically highlights huge differences in the migration strategies of puffins from different colonies. You had colonies like the Gulf of Maine, for example, which is in sort of green-blue, that didn't go far at all. They sort of stayed near the colony the whole time. Or you have long-distance migration like the pink birds from Iceland, 4 colonies in Iceland, that all went to the Labrador Sea and then southeast. You can see in red the Isle of May birds staying mainly in the North Sea, and then in purple and orange you have the Norwegian birds, using the Barents Sea and then going towards Iceland as well. And so this really allowed us to pinpoint the important areas for puffins in the non-breeding season and highlight also areas which were used by multiple colonies. And then we scaled this by the population size of these different populations to obtain this map. And you can see here that really important areas for puffins in the winter are around the Labrador Sea and then in this area sort of south of Greenland. What was really nice is actually that data was then used alongside other tracking data by BirdLife International to propose a new marine protected area in this area, in the North Atlantic, which has now been accepted by Swan, is now a marine protected area in the high seas. And I forgot to put the reference, but this was led by Tammy Davis and was published in 2021 in Conservation Letters. But because we had this great data set, tracking data from 13 different populations, we wanted to take advantage of this to try and understand what was driving these different migration strategies in populations. And I'm not going to go into all the details of what we found, but there's 2 key results I want to mention. The first is that we found that migration distance could be predicted by 2 factors: colony size and the local winter habitat quality. So puffins from larger colonies and from colonies where the habitat quality around the colony in winter was rubbish migrated further than puffins from smaller colonies and with better habitat quality around the colony, which kind of makes sense. But basically, this is quite a neat demonstration, potentially, of the role of competition on migration distance. We also found that these migration strategies and how tough a time puffins were having in the winter was reflected in the productivity the next year at the population level. So puffins that had to migrate further or who were wintering in areas which were less productive had a lower breeding productivity. And there was also an influence, again, of colony size and latitude. And that's also quite a neat result because it shows that if you want to understand, for example, a dip in productivity in a colony, you can't just look at what's happening during the breeding season. There is really a need to incorporate the whole annual cycle, and obviously what's happening during breeding is key to breeding success, but there are other factors, and what's happening during migration definitely can also play a role. One thing that we didn't really look at, and I don't have an answer for, but I'm quite interested in, is that some of these populations had this huge route diversity, like I showed on Skoma, with different puffins were going to completely different places. And some populations all the puffins were kind of doing the same thing, like Iceland, for example. And I don't know why that is. Perhaps it is because the populations, one idea is that the populations on the edge might be slightly more exploratory, and so you end up with more diverse migration routes. I don't know, but this is something that we haven't really looked at yet, but that I find worth mentioning because it's quite interesting. Okay, so we're leaving puffin behind. These drivers of migration strategies may also vary between environments, and this is something that I've been started to investigate more recently. And for this, we're going a little bit further south, and I'm taking you to this incredible place called Aldabra Atoll in the Seychelles. That's in the Western Indian Ocean, kind of north of Madagascar. Aldabra Atoll is the world's second largest raised coral atoll, and it is incredible. It is home to an amazing array of species, and it's just teeming with wildlife, especially marine wildlife. If you walk into the waters up to your knees, you'll have sharks and turtles zooming past, and fish from lots of different species. It's got lots of mangroves, which are really important as shelters for the young turtles and fish, and also birds. And it's also got really interesting and important terrestrial wildlife, in particular the Aldabra giant tortoise. And there's nothing quite like being woken up in the morning by the sound of giant tortoises mating in front of your bedroom. It's got also lots of endemic land bird species, and it's also very important for seabirds. It's the world's second largest colony of frigate birds. It has got a lot of red-footed boobies. We don't know how many yet, but there's a lot. And it's got lots of other species. These are just a few: fairy terns, noddies, and tropic birds. And we don't really know much about any of these seabirds, actually. And that's because, they haven't been really studied. So Aldabra, Seychelles used to be a British colony. And then while it was still a British colony, in the sort of 1950s or 60s, the UK government had decided that it would be a perfect place to build a Royal Air Force base in the Indian Ocean. And so they had plans to basically blow up the channels of this island and install a Royal Air Force base there. Luckily, the plans were leaked to the public, and there was a massive uproar, especially from a lot of scientists worldwide, highlighting how it would be disastrous because this place is really a unique biodiversity hotspot. And so thanks to this pressure from the public and from scientists, the plans were then abandoned, and the base was constructed on Diego Garcia and the Chagos instead. So what happened to Aldabra instead is that the Royal Society organized an expedition in the 1960s with a bunch of scientists, and the person in charge of studying the seabirds was Tony Diamond, who's now in Canada, but at the time was doing, I think, a postdoc at the University of Oxford. And so in the 60s, he went there and sort of described the seabirds of Aldabra and their breeding ecology. And these papers were sort of published in the early 70s. And then there was some research on Aldabra, but no one looked at the seabirds, and now that Seychelles became independent in 1976, it's now managed by the Seychelles Islands Foundation. They have research staff there all year round, but they're mainly focusing, and this is now luckily changing, but they've been focusing mainly on the marine wildlife and the tortoises. And so really there's not been studies of seabirds in Aldabra for a very long time. And when I went there, I went to study this bird, the wild-tailed tropic bird. The main purpose of my work was to look at the foraging movement, but I'm not going to talk about this today. I just wanted to show you a chick. They're amazing. They're like super cute little fluff balls. And this is just a video of, this is very sweaty me, in the middle of the lagoon. And this is where the tropic birds breed. So there's this huge lagoon, 50 kilometers wide, and there are rocky islets in the middle. You can see here there's basically dead coral, and it's very dense vegetation on top, and the tropic birds breed under this vegetation. And you can see the amazing clear water, which is very good when you want to see the sharks and the turtles around. You can see here this little orange tape marks a tropic bird's nest. And we basically every day have a 2-hour window at high tide to get to these islets and study the tropic birds. Here's a tropic bird nest, and you can see the egg there under the bag. And this is a red-tailed tropic bird, actually, which is another species I was studying there, that I'm just going to put back on the nest after taking a tag off. So I feel very privileged to have been working there, and all this work was done in very close collaboration with the Seychelles Islands Foundation. So although I went there to study the foraging movements of tropic birds, I also put some geolocators on the white-tailed tropic birds. And this was actually quite recent, so this is ongoing. And we're getting every now and then some geolocators back. So I don't really have a formal analysis of data to show you. I'm actually just going to show you 2 tracks that we've received so far. But they're really interesting. So this is a bird, white-tailed tropic bird, that we tagged in December on Aldabra. Aldabra is the little white square. And it was breeding. And in January, so just a month later, it failed. And so what it did is it went on a migration, you could call it that way, for about 4 or 5 months, all the way to June. And these are the yellow dots on the map. And so it went eastwards and then came back to Aldabra. And then in June, it started breeding again, because white-tailed tropic birds in this tropical environment can breed at any time of the year. So it started breeding again in June, but then failed again in August. And then it did a second migration for the year, again, just 4 or 5 months. And this time it went to a different place. It went further east, and then came back again, started breeding again. This is when we caught it. And so this shows, first of all, that what we think about what migration is might be very different in these tropical species, which have a much more flexible annual cycle, and they can breed at any time of the year. It looks like this bird has decided to have 2 migrations for about 4 months after every time failing to breed. And it also looked like it went to 2 different places during this time. Now, this is in contrast another tropic bird that was also breeding in December when we tagged it, but this one was successful. And so it was breeding all the way till April. And then it took just this one migration that was all the way to the next December. So this is much more similar to what we'd see in a temperate species. And this bird actually went to roughly the same place as the first bird did in its first migration. Now, this is just n equal 2, and we're now getting more data, so we can now analyze this more formally. But I think what this highlights is that what's driving migration strategies in these species will be quite different from what's happening in temperate species or polar species. And this might really have an impact on their annual cycle and also the sort of how carryover effects work in these systems. And this is really something I'm looking forward to looking into it soon. So this was a really strong demonstration of how breeding can affect subsequent migration, in this case, duration and also destination. But to disentangle sort of more subtle effect and disentangle this from also individual quality, it's better to use experiments. And so we're returning to SCOMA to look at an experiment that I've done, this time not on puffins, but on Manx shearwaters, which are much easier to work with, and especially for experiments. And before we go into the details, I just want to give you a little insight into what it's like to be on the world's largest Manx shearwater colony at night. And this is footage that was recorded by Matt Wood with a thermal camera a few years ago. It's just the most fantastic experience to stand on this island in the dark and have tens of thousands of shearwaters just flying past, and just the sound and it's amazing. So I highly recommend the experience if you want to visit SCOMA. So anyway, let's go back to the experiment. So the experiment aimed to manipulate the cost of breeding in these birds and then measure how this affected subsequent migration. So to do this, we swapped chicks between burrows. And that's something we can do in shearwaters because they don't recognize their chicks. So if you swap them, they don't really have a clue and they keep feeding them, which is really handy. So we had a treatment group that was the lower effort group, and they already had quite a small chick, and we replaced it with a chick that was approximately 20 days older. And that means they didn't have to feed it for as long as planned. And then the higher effort group was the opposite. They already had quite a big chick, and they received suddenly that chick went down to a small chick size that was about 20 days younger. And the idea was that they might have to feed it for much longer than planned. We also had a control group that received a swap chick but from the same age. We weighed the chicks every day so that we could follow their growth rate, and we also put some geolocators on to look at the migration of the parents. So this is the timeline of the experiments. We did the chick swapping, we weighed the chicks, and then we followed the adults for a year, including the next breeding season, because we wanted to look at their behavior then, and also looked at the chicks the next breeding season. And for a subset of birds, we also had sort of control data, which was geolocated data from a previous year without any experiment. And what we found is that there was definitely an impact of this manipulation on the migration of these birds. Now, I should say, among shearwaters, unlike the puffins that were going all over the place, all the birds from Skomer, and actually from many other colonies in the UK, winter towards the Patagonian shelf, so they all have very similar routes to the Patagonian shelf and back. Now, the bird that had an easier time, the lower effort treatment, indeed had to feed their chick for much less long, so their season ended earlier, they didn't have to bring as much food to the chick, and they spent a lot less time foraging. And that allowed them to start their migration significantly earlier than the controls. Now, in contrast, the birds from the higher effort treatment, we thought perhaps they'd just abandon their chick and start the migration, but no, they stayed until the end, fed the chick, and so they were forced to start their migration significantly later than the other birds. And that meant they got to the wintering grounds later. In terms of behavior, we saw some small differences in behavior at the wintering ground, and especially the lower effort treatment, who had an easier breeding season, spent more time resting. And then, interestingly, all the phenology reset at the start of the spring migration. They all started migrating again, all three groups, at the same time. And so we thought, well, maybe that's it. Maybe they've recovered, and there's now no more differences. But actually, there was the next breeding season. The sample size now for the lower effort treatment, for which we had data for the next season, was quite low, so we didn't really find a significant difference, but there was a trend for these birds to do a little bit better than the controls in terms of lay date and chick mass and so on. But for the high effort treatment, they did significantly less well than the other birds. They laid later, they laid smaller eggs, their chicks were lighter, and overall the breeding success was significantly lower. And so what that means is that even a small increase in the cost of breeding 1 year could still be felt an entire year later in these birds. And that's really interesting and really important, because it means that we're now in a context where a lot of seabirds are going to face hardship, maybe multiple years in a row. And that means that these effects may actually accumulate because they might dissipate really slowly. And this is something I've started looking at last year. I went back to Skomer and we did this experiment with more treatments where we manipulated the cost of breeding at different times and after breeding. And I'll be looking at these birds over a longer period of time to try and measure how long these effects take to dissipate. So there's one key aspect of migration I haven't mentioned yet, and this is how seabird migration routes develop. There is now growing evidence that the common established theories of migration routes inheritance in birds may not apply to all seabirds. Now, these common theories are either genetic inheritance, you inherit genetically your migration routes from your parents, and so it's inherited as soon as you left your nest, you know where you're going. And this is, for example, the case in many passerines. Or social inheritance, where you might just follow your parents or a family group on their migration for the first few years, so you can learn the migration routes. And that happens in, for example, some storks and some geese. But for example, in the case of the puffins on Skomer, this is really unlikely that this is what's happening. The sheer diversity and complexity of these migration routes mean it's very unlikely they're genetically encoded. And also, pufflings fledge at night on their own, and the parents stick around the colony for another few weeks, so there's no way they're migrating with their parents. And so based on this puffin data, Tim Giffen came up with this potential third theory for how migration routes might develop in some seabirds, which he called exploration refinement. And this is basically the idea that young seabirds might just explore different routes in their first years, in their early life, and then gradually refine these routes as they grow and as they experience more places, so that by the time they start breeding, they then have their own migration route. And this would explain why we have different puffins going to different places, but each puffin keeps going to the same place over and over again. But that's actually quite hard to test. So this kind of makes sense in seabirds because they have this long breeding deferral period, and so there's really scope for these birds to explore and to learn during these first few years, and also for variation to arise between individuals. And there is a little bit of evidence, even if you just look at some images, that there is definitely some learning going on here. So this is just a video, sorry, that might be a bit loud, of some tropical shearwaters on Aldabra, and these are immatures. And basically, they just spend all their time on the colony in the evening. They don't go into burrows or anything. They just spend all their time fighting and shouting at each other. And there's also a lot of preening. They would spend hours and hours, just all night long, just preening each other and also practicing to mate, which you'll see in a second. They haven't quite worked it out yet. And so these age-driven differences are really difficult to study because of the elusiveness of immatures. But they're really important because actually immatures might represent quite a substantial chunk of a population. And so if we want to, we need to understand the behavior and the distribution of these birds, especially if they are of conservation concern. Now, we had a little go at doing this with mixed success. And my very first year on SCOMA in 2011, we deployed geolocators on shearwater fledglings. And actually, we didn't do this on SCOMA. We went to Copeland to do it, which is a small manx shearwater colony in Northern Ireland. And we didn't do it on SCOMA because there's so many shearwaters that the likelihood of finding a chick coming back of the geolocator is pretty much zero. Whereas Copeland is a much smaller colony, so it's more likely to recover them. So we did this in 2011, and then over the next 5 years, we only got 3 tracks back. And then more recently, Joe Wynn, who is here today, and he's now a postdoc at the University of Oldenburg, but he was doing his PhD at the University of Oxford, has now led the analysis of this data. And what we found from these 3 birds, even though it's a very small sample size, is very interesting. And you can see the 3 tracks on the left-hand side here in color. And what we found is that basically, as soon as these chicks left the colony, straight away, they started their migration. There was long periods of flights from the beginning, and they headed straight to the Patagonian shelf in Argentina. And the black points that you see behind the colored dots are the adults. And so really, they follow pretty much the same route as the adults, and they do that immediately. Joe also put some transmitters on some fledglings. Unfortunately, it didn't work really well, so he only got a little bit of data. But this also confirmed that these birds are just going south immediately, and that's on the right-hand side. And because that data is more accurate, we could also validate that they're not migrating with their parents. They're not following their parents to Argentina. And so this might be quite good evidence that actually there is some genetic inheritance here. And so we might think, okay, well, we've solved the development of migration routes in shearwaters. Well, not quite, because actually what happened next was really interesting. The first geolocator had already stopped working by this point, so we ended up with only 2 geolocators. So the birds had arrived around November in the Patagonian Shelf area, like the adults, and they wintered there with the adults. But then the adults started coming back to SCOMA around sort of February, March time. But these birds stayed longer, and they didn't start moving north until July. So they had stayed much longer than the adults. And these are the blue dots. And when they started moving north, they only went a little bit north, all the way towards like sort of the coast of Brazil. And then they stayed there for a few months, and then they came back down in September. And then the adults joined them again after breeding around October, November time. So that was the first year. And then the geolocator on the left then stopped working. So we only have 1 more bird for the end. But that's when it becomes even more interesting. So this bird then, in May, so a little bit earlier than the previous year, started moving north again, but this time it went properly north. This is the orange route, so it goes all the way, and it follows a very, very similar route that the adults use when they go back to breed in spring. So they go along the coast of South America, and then towards the Caribbean, and then and then they cross the Atlantic from the US. The bird didn't go all the way to its colony. It stopped about 500 kilometers away, and that's not really surprising because we don't really see first years at the colony. And then interestingly, what's interesting is the way it went back down. So it basically retraced its steps all the way back to the migration, to the wintering ground. Now, we've been tracking hundreds, perhaps even thousands of adult manx shearwaters in lots of different colonies in the UK, and we've never seen a bird do that. All the adults migrate the way the fledgling did when they left their nest, that is going south along the coast of Spain, Africa, and then cross and go to the wintering ground. So this was really surprising. We really didn't expect to see this. And then the bird did it again the next year, and then suddenly the geolocator stopped in May when it had almost reached the colony, so we don't really know what happened next. But this shows already that even though this first bout of migration seemed to be genetically inherited and very similar to the adults, then there was something completely different happening in the next couple of years. Now, again, this is a very small sample size, so it's hard to make very big conclusions, but already this raises very interesting questions. And when I said we never saw an adult doing this migration, it's not quite right, because a couple of years later, I ended up on Martinique's Rock, which is a small island in Maine, in the US. And there were no there's very few Manx shearwaters breeding in this side of the Atlantic. But around 2010, I think, researchers on Martinique's Rock, the researchers are from the National Audubon Society in the US, they started hearing Manx shearwaters flying past at night, but not landing. And then as the years went by, a few landed, and then they had their first breeding pair. And at the time I visited, they had 5 breeding pairs. And we got permission to deploy 2 geolocators on these birds. And these 2 birds followed the migration route that you can see on this map here, which is what this immature bird had done. So not the way that the British birds migrate south. And this is actually really unfavorable wind condition to go south, especially for the first part of the migration. And this is quite interesting because this colony is growing, and we don't know where the birds are coming from, but to be fair, we think it's more likely that they're coming from the UK because there's not really sort of source colonies around that they could migrate from. And we're just wondering whether actually these birds discovered this colony on these return migrations and then started a new colony there. So we'll be keeping an eye on them, and once the colony grows a little bit more, we'll be able to do a bit more tracking, and we might also look into doing some genetic analysis to work out where these birds are from. I also tried to do the same chick tracking experiment in puffins. When I started my PhD in 2012 and 2013, I deployed some geolocators on puffins because I wanted to know how this incredible diversity of routes arose. And this didn't work really well. In particular, the timing was pretty poor because in winter 2013, there was huge storms in the Atlantic, and there was a massive puffin wreck, 30,000 dead puffins washing up on the French coast, and a lot of them were from Skoma. So actually, that must have killed off a lot of these chicks. Their breeding survival is already not very high in the first year, so this was not a good timing. And this is actually the only track I got back is from a dead chick that was found on the French coast. It was by then 1 year old. There is another bird that exists, that's still alive, and that is that bird there on the right. And this is now my nemesis bird. It has returned to Skoma, and I know it's one of them because they have this grey ring, and we don't put the grey rings on anyone else. And it's not been breeding for years, and so I've never managed to catch it, and it's getting closer and closer to the edge of the cliff. And I'm not sure I will ever manage to get it, but every year I can see it. And so 50% of the data available in this experiment is on this bird. Maybe one day I will get him. So this is what this one track showed us. Basically, this bird left Skoma in July 2013 as a chick, and then as a fledgling, and then it went for a few weeks down into the Bay of Biscay, and then it spent pretty much like several months west of Ireland, and then for the next year, it kind of made its way west in the Atlantic, and then a bit north towards Iceland, and then back down. And then in January 2013, 14, I can't remember, 14, it was hit by these big storms that killed so many puffins, and it died where you see the little triangle, and then it was recovered on the French coast about a month later. Again, this is a tiny sample size, so we can't really say much, but it doesn't look like what the fledgling shearwaters are doing, which is heading straight to one destination. And this might actually, once we have tracked more birds, this might provide support for the exploration refinement hypothesis. Now, luckily, SEATRACK have now started to deploy lots of geolocators on seabird chicks of multiple species, and so we might get insight into this system soon. And I won't say any more about this because Birger will talk about this in the next talk. But there is additional support for the idea that young birds learn and explore in these first few years, and that comes from data we've collected during the breeding season. This was work that was led by Steve Vautier, and he was tracking gannets on Grasholm next to Skoma. And he tracked adults and immature gannets, and he found that the immatures, which you see on the right, were much more exploratory than the adults. The adults had high route fidelity and return feeding to the same place, whereas the immatures were going all over the place. And so this might suggest that this foraging specialization of gannets is learned during early life, and actually maybe that's why they're deferring breeding for so long. Maybe seabirds have to learn how to migrate, how to feed, and how to do all these things, and it takes years to do it, and so they only breed when they're ready to do it. But are these birds actually learning to feed? But to test this, I tracked some immature mangrove shearwaters on Skomer, and this was quite a while ago, so we didn't have remote download tags at the time. So what we did is we put some I-got-used on these immatures and then just sat there all night for weeks, waiting on the colony in the dark to see little blue flashing lights, which would indicate these birds had come back because they don't go into a burrow, and they could just land anywhere, and then slightly crawling up to them to catch them when they arrived. But it was quite fun, and we did manage to track about 20. And we found that there was quite a lot of spatial segregation between the adults that we tracked at the same time and the immatures, as you see on this map with the immatures in orange. We also found that the immatures were foraging less efficiently, so the amount of mass gained per unit time foraging was significantly lower than that of adults, and they also forage in less productive area. Now, this could be because they're displaced by competition from the productive areas by the adults, or perhaps they are just not good at finding the productive areas. So, yeah, that we don't know. Okay, so I'm going to end there. Just to conclude, we've seen that seabird migration movements can be shaped by many factors, and it's quite a complex story to get our heads around. And these factors act at the individual and at the population level, and that the variation in these strategies can definitely affect fitness, breeding success, and ultimately population trends. We've learned a lot about seabird migration in the last few decades, and we know a lot more than we did before, but there's actually still a lot more that we need to understand, and especially the links between migration and other events in the annual cycles, carryover effects, and how this thing integrates over the life of the bird. And we also have a lot to learn about how this works in different environments, especially when seasonality is different. And ultimately, that's important because it could affect how seabirds will react to environmental change, which is becoming, obviously, a really big concern. Thank you very much for listening, and I'd be happy to answer any questions.
Excellent talk there. We have time for quite a few questions if people's brain power is up to it this morning.
Hi, Annette. Thanks. That was really interesting. I wanted to ask about the puffins pairing with partners with similar migration routes, and then how you think kind of that happens.
Yeah, that's a really good question. I don't really know, because we did think if, is there potentially associative mating that happens at sea? But it seems very unlikely, especially now that we know the migration patterns of these birds, that they would end up meeting a mate at sea. And we see a lot of courtship happening at the breeding site. That's quite unlikely. I wondered if actually they meet at the colony, but if they are already migrating to similar places, they might come back to the colony more at the same time, and so they're more likely to meet a mate with the similar routes. Or maybe this happens after mating. Honestly, I don't know, but yeah, that's a good question.
Hello. I was wondering, with the exploration and refinement, because all the colonies are going to slightly different places, do you think while they're exploring they kind of follow others from the same colony?
Do you mean other young birds?
Yeah.
Yeah, potentially. I think that sometimes when actually you go just around the colonies, you can see there are, little groups of fledglings. But I think there's not really much evidence that they do lots of group migration. So I think it might be quite a lonely thing that they do on their own. But then actually, I'm saying this for Skomer, but it's for the colonies where, like Iceland, for example, where there's much less differences in the migration routes of adults, it might be possible that there's less exploration also in the younger birds, and that they just end up all going the same way. But I think, I reckon it's probably something that they do individually.
Fantastic talk, thank you very much. And a rare example of a manipulative experiment. We don't see that much in seabird ecology these days. You can go through a whole conference with maybe only 1 or 2 experiments. And given your take-home message about connections between seasons, fitness consequences, carryover effects, should we as a community be doing more experiments?
I think we should, because I think we can learn, and we have learned so much from observations. But for really complex systems, especially when there could be lots of different factors at play, if you want to disentangle things, experiments are a really neat way to do this. But it's also not very easy. So, for example, shearwaters are quite easy to work with, but there's loads of species. I wouldn't even dream to do this in puffins. I think it would be really difficult. But also there's the ethical implications of doing this. So, for example, in this experiment I presented, I did increase the cost of breeding for some birds, and this has to be justified to an ethical board, and it might not be the right thing to do. In my case, I only wanted to have a small effect, and so this was accepted. I wasn't trying to do something big, but this is also an issue with experiments. But I absolutely think we should be thinking more about experimental design, experiment in seabirds, definitely.
Perfect. We have one more down the front. Was it you?
That was a really good talk. The partners that kind of seem to do similar things on the outward migration for the puffins, is it possible that it's just geolocation error, or are they different enough and distinct enough that you can be confident that they're doing different things?
You mean the different adults migrating to different places?
The partners. So,
the partners.
Tracks. And the tracks look quite similar, but they were just kind of offset by a small bit. And is it possible that it's geolocation error, or is it kind of different enough that you can be confident?
Yeah, there is definitely some geolocation error, but I don't think this result is just purely based because of that, because the difference is quite significant, and actually there's geolocation. You wouldn't expect the geolocation error to go all into one direction, so it would also maybe make close tracks more different. So I think the result was different enough that there is something there. But yes, it's always the problem with all these geolocated studies.
Because the tracks look quite similar, so I was wondering if it's slightly offset because it's
Yeah.
like angles or so.
No, I think some of this similarity was really clear in the data, and also we had we've since then got much more data that also confirms this. So yeah, I think it's not just there.
Really good. Thank you.
Thank you.
Perfect. Oh, thought you'd get away with no more questions, but sorry.
Good, fantastic talk. How long do you think it would take the Manx to fully recover from the chick experiment? So when they got the younger chick?
That's a good question. I don't know, and that's why I'm trying to find out now by trying to look at this in the longer term. So there was definitely still an effect a year after. I would hope that maybe, I think 2 years is probably, if they have a standard breeding season the next year and the conditions are not terrible, maybe 2 years. But that was only a very small change. So obviously it also depends on the manipulation. But honestly, I don't know. It looks, so what's interesting is it looks like the positive manipulation, so when we made it easier for them, didn't have as big an effect as the negative one. So actually maybe, which is a bit of a shame, because maybe when they have a bumper year, that doesn't lead on for so long, and this maybe is the negative stuff that lasts longer. But it's also difficult to study them over a very long time, because your sample size keeps going down, because there's always some birds not coming back, or some geolocators stop working, and so it's actually quite tough to go very long in the future. But hopefully looking at 2 years will at least give us a better idea of how long that stuff lasts.
Perfect, thanks. I think we'll give Annette a break now. She's answered enough questions. If you have any more, I'm sure she'd be happy to answer those during the coffee break. It does give us now an opportunity to move slightly ahead of schedule, which means earlier tea break, and we might kick off the rest of the talks here for the session. Jamie, could you help? Thank you very much. So the first talk in this session is Hallvard Strøm, looking at large-scale tracking of seabirds in the North Atlantic, featuring the large-scale SEATRACK program.
Good morning, everybody. Paul, can you hear me up there? Okay. Yeah. So, sorry to say that Hallvard couldn't make it to have this presentation, so it will be me having this presentation. The good news is that the organizers, they have discussed to give you the money back for the conference fee. Is that right? Yeah. So I will be having this presentation on behalf of the project group, say a little bit about what is SEATRACK and design and approach of the program, give you a little bit of a status, results, and say a little bit about how we foresee the next phase of 4 years for the project. The main goal of SEATRACK is to map the non-breeding distribution of seabirds breeding in colonies encircling the North Atlantic, and also to look at how changes in environmental conditions in wintering areas affect their demography and population trends. So SEATRACK has a science component with a science plan, and we also have a management component with specific deliverables and products that we have promised for management and those funding the project. So the phase 1 of SEATRACK started in 2014, and at that time we had the geographic scope was in Northeast Atlantic, and then in 2019 we started phase 2. We expanded the geographical scope to the northwestern part of the Atlantic to include Greenland and Canada, but we also increased the range and the scope coverage in specifically Ireland and Scotland. Well, phase 2 is ending this year actually, so that's why we are also have been working with the plans and application for a next 4-year phase, the phase 3. And so with such a multi-colony and large-scale project, we certainly depend on large-scale collaboration. So there are more than 60 scientists involved and partners from 10 different countries, and many are here, of course, today. SEATRACK includes 11 seabird species from 4 ecological groups, kittiwakes and fulmars from the pelagic surface breeding, feeding birds, glaucous gulls, herring gull, and lesser blackback gull, coastal surfer feeders, while common guillemot, British guillemots, Atlantic puffin, and little auks are pelagic diving birds. And finally, common eider and European shags are coastal diving seabirds. What is important with the SEATRACK design and is the infrastructure and the common protocols of the project, so that all the field teams are following the same procedures. There's also additional sampling, and we collaborate closely with Arctox, which is then linking samples and measurements of contaminants to the non-breeding movements. Very important for SEATRACK is that we have a centralized and standardized processing, so all the GLS data is processed in the same way. The new data coming in also with the old ones. And if we change the procedures, we update all the old data with the same procedure. All this is put into a centralized database and a file archive, so when we need to use the data for management purposes or scientific purposes, it's efficiently extracted from the database. And we also have an agreement of understanding between the partners and SEATRACK because of shared data ownership. And so when somebody wants to use the data, there's a data request form that will be circulated among the partners. The science plan has 4 research themes. The first one is about variation in migration strategies among individuals, populations, and species. The second is a linkage of migration strategies and winter distribution to demography and population dynamics. And we have one theme on linking non-breeding distribution to contaminants, and the final one is about marine spatial planning. And so far, SEATRACK has contributed to more than 30 papers, and the most recent achievement is the theme section in Marine Ecology Progress series. This one, where 12 original research articles fill the entire theme section, and we're really happy about that, of course. And if I brought a few copies of this one, so if any of the partners wanted a copy, they can have one. So this obviously depends on a lot of good data, and since 2014 we have deployed more than 15,000 loggers on this species. We have retrieved over 7,000 loggers until 2021, which is quite good. There's still more to come in this year. And when we plot the filtered positions from the database on a map, it looks like this, with more than 2.5 million filtered positions and more than 7,700 tracks and 3,600 individuals. And this doesn't show the pre-SEATRACK data, but this is also, so if there's data from before SEATRACK, before 2014, we also accept that, and we reanalyze and process them the same way as the SEATRACK processing, so that they can be compared and used the same way. And these data are also, these old data, pre-SEATRACK data, is also included in the first map product we have, which is kernel maps, where you choose to show, so it's a web application available at that address shown below there. You choose the species and the season, so you can choose between autumn, winter, or spring. You choose whether you want a specific year or all years, or a specific colony or all colonies, and you have maps like this. So this is all the data we have for kittiwakes shown as kernels during the winter. As the second map product we have recently developed is the population distribution map. So Per Føkal has made a data set called NEAS. So what he has done is he has combined population size data with the tracking data and also habitat modeling. So the idea, we don't track birds in all the seabird colonies in the geographical area, but we let the tracking data represent neighboring colonies as long as they're not too far away. And by doing that, this approach can represent 87% of the North Atlantic populations. So the way to increase this coverage is, of course, to have a better representation of deployments where deployments are lacking. This so far includes 6 common pelagic species, but there's ongoing work, so 5 more species, the more coastal ones, and also the Northwest Atlantic, Greenland, Canada, will be included in the next developments for winter now. And this we have made a new web application. It's a prototype available that will be tested among the partners now, so the partners will be able to test it. It's kind of the same approach as the previous one. You choose the species and choose the ocean area, choose land area, and so on. And here you can clearly see which colonies are included and not. So this is an example where you see the blue ones are colonies that are not, they're too far from the nearest SEATRACK colony where we have been tracking, so they can't be part of the calculation. So try it out, and we can have some feedback. Okay, so for what do we foresee in the next 4-year phase, the phase 3 of SEATRACK? We think we would like to keep the main goal and the geographical focus as in phase 2. So we stick with the North Atlantic. However, we would like to do some assessment and to assess the potential for collaboration with the Pacific, because we already have shown that kittiwakes from Novaya Zemlya, they migrate across the Arctic Ocean and winter in the Pacific. And with the ongoing climate change, the sea ice barrier in the Arctic Ocean is getting thinner and weaker, and by 2035, the climatologists have predicted that there will be no sea ice during summer. So this issue is becoming more and more important, so we will do some assessment of this potential for collaboration. We will want to continue to develop the map products and add another one about vulnerability. And also, even though SEATRACK is already a very large project, it only covers a few number of species, and we lack some seabird groups. That's why we started at the workshop in 2021 with our partners to discuss criteria for including potential new candidate species, and we have continued that work, and we have then based on biological and practical criteria, come up with 5 candidate species so far that we will discuss more closely with our partners. It's the Arctic tern, great skua, northern gannet, leech storm petrel, and razorbill. So we certainly want to get in touch with teams working on these 5 species who are interested in joining the project. We have, of course, made a list of potential sites where this can be feasible, but we are definitely interested to get in touch with you. And a very interesting development now is the work we've been doing on whole year tracking with GPS, foot-mounted GPS loggers. So it's a collaboration with SEATRACK. They have been working some years on this now, and finally we managed to get the right devices to try them out this year. So this year we tried out foot-mounted GPS devices from SEATRACK, about 4 grams. It looks like a big, you can see it on the leg of the glockenspiel there, it looks like a big GLS logger, smaller than some of the TDR loggers that have been quite widely used. But you also can, if you see, well, you can see there's a small antenna. It's a small, quite flexible antenna also. So with this one, of course, we can avoid some of the disadvantages of the GLS logger, so we can avoid the problems of equinox, but also midnight sun and polar night is a huge challenge for a program which is centered so far north. So far we tested this in 5 species in Bjørnøya, Kongsfjorden, and also at Isle of May before the flu influenza got too problematic there. So it's a start. So we are excited about how this develops and whether the technology works well enough. Annette introduced the concept of the young birds. They are a really important part of the seabird populations, but we know very little of their movements. Sometimes this period from fledging to recruitment has been called the lost years. We will certainly try to expand this in the next phase. So far we've been working on the 6 species in the northern colonies. I only show one map here of young glaucus girl. So clearly you can see an example with the orange kernel field, which represents the wintering area, that it does something else than the adults. The adults are not shown in this graph, but they are certainly much further to the south during the first winter than the adults. And then they gradually do more adult-like wintering behavior. Okay, so there were only a few results presented by me. There will be more by Benjamin, the next talk, and he will provide some excellent examples of how data from SEATRACK can be used. Also thanks to the organizers and the funders and the partners, of course, and to all of you for listening. Thank you.
Thanks very much for the overview of a huge project with great potential for collaboration. Are there any questions on this from members of the audience? Nobody's keen to be involved in this large-scale collaborative project, I see, no? I'll kick off with a quick one. Obviously, this has been a very large-scale project. How has this been funded, and how do you anticipate the continued phases of this being funded?
That's, of course, a good question and a big challenge. So far, it has been... so to start with the base of it, it wouldn't be possible without the funding of CPOP and all the other participating partners who are doing long-term work in the colonies. So that's a big base of the project. On top of that, there's funding from the Norwegian government and then from industry partners, mainly from the oil and gas industry, but we also try, but they also go now into wind energy, but so far it hasn't been much from the wind industry yet, but mainly oil and gas partners. So this is of course also a challenge if we want to expand in the next phase, that we also depend on having a larger contribution. And so far, the contribution from the other countries has been the work that has been done in the colonies.
Perfect, thank you very much. Any questions from anyone else in the audience, or will we get on to the next talk where we get some more detail? None? Just running myself this morning.
I'm wondering, with the immature tracking, because the return rate is so low, do you have a specific strategy to try and maximize how many birds you're getting, or is it just deploying a ton of loggers and hoping for the best?
So it was a bullet point on the slide that I forgot to mention. So an important part here is to try to ensure that we have no device effects. So we are now trying to do an experimental approach to include the control groups that are being captured and treated exactly the same ways as the young birds that get the logger, and then to compare return rates and so on. And with that approach, we can't be too selective. Then we have to be more random on which birds we deploy and not, so that will be a random process. But to ensure a good return rate, the most important thing is to have people in the colonies with a very high attendance and to scan and scan and scan, like you show pictures and you wait and you look. So it's a dedicated teams and topographic features of the Seabird cliffs are kind of maybe the most important thing. Yeah.
Wonderful, thank you very much. I'd like to thank you.
Hello everyone, morning. I'm Vicky Warwick-Evans and I'm a postdoc at the British Antarctic Survey, and I'm going to be talking about our Darwin Plus and South Georgia government funded project looking at the spatial segregation of wandering albatrosses at South Georgia. So firstly, thanks to Lizzie who gave a great introduction to wandering albatrosses yesterday, so I'm going to move straight into what I've been doing. But seabirds are amongst the most globally threatened birds, as I'm sure a lot of us know, and this is often as a consequence of bycatch in fisheries. And many of the albatross and petrel populations breeding around South Georgia are in decline, and the rate of decline varies quite considerably amongst these different populations. And we think that this might be to do with different interactions with the fisheries whilst at sea. But until this year, the only tracking data of flying seabirds had been from birds breeding at Bird Island, and we think that this may have implications for conservation and management. So you can see from our plots here our study location. In the top plot we can see South Georgia in relation to South America and the Antarctic Peninsula, and then underneath we've got our two study locations. So at Bird Island we've got a long-term field study site, and from this we were able to deploy high-resolution GPS trackers that we could put out for a single foraging trip, recover the device, recharge it and redeploy it. But at Pryon Island, we were able to access the site for one day, so we put out PTTs that are basically satellite trackers that ping a location approximately once every hour, and you don't need to recover the device to get any of the data back. So from both colonies, we focused on breeding adults, and we collected data from January to July this year. But for the purposes of this talk, we're just going to focus on incubation, which is January to March. We also put some geolocators out to track during the non-breeding season. On Pryon Island, we deployed these on non-breeders in the hopes that they'll be coming back to breed this year and we'll be able to recover some of them. And at Bird Island, some were deployed on breeders and some on non-breeders, so we do have some of this data back already. So the analysis I've looked at so far has been, firstly, although we had this nice high-resolution GPS data, unfortunately our PTT data was much lower resolution and we wanted comparable data sets. So we've used crawl just to make a regular 1 hour data set for each of the two colonies. Then we've used kernel density analysis to look at the 50% and 95% core foraging area and home range area. Looked at how the populations overlap using Bhattacharya's affinity. and then trying to understand potential causes of the variation in these at-sea distributions, looking at the wind direction when the birds leave the colony, done some habitat modelling to see if they're foraging in similar habitat types, and finally use some of the Global Fishing Watch data to look at the overlap with fisheries. So firstly, going straight to the tracks, the top plot here is from Bird Island, and we can see that there are some distinct areas of similarity, but also some distinct differences with those tracked from Prion Island. So both populations tend to use the locations between South Georgia and South America and up onto the Patagonian shelf. But birds from Bird Island also tended to head down to the shelf area by the Antarctic Peninsula, as well as further over to the west, whereas those from Prion Island tended not to use the area by the Antarctic Peninsula, but to travel further east over past the South Sandwich Islands instead. So just having a quick look at some of the summary statistics, in all of the plots, Bird Island is green and Prion Island is blue, and we can see that there doesn't really seem to be much variation in the trip duration, except for this one outlier from Prion, who travelled for over 800 hours on one foraging trip. From the maximum distance from the colony also doesn't seem to be particularly different, except for these two birds from Bird Island who went very far west. And again, the total distance travelled doesn't seem to be hugely different either, except for another outlier from Prion. So looking at the kernel density analysis, we can see that for the 50% core foraging area, the locations the birds use from Bird Island sit exclusively within the area of those birds from Prion Island. But then looking down at the 95% home range area, we can see again this distinct overlap between South Georgia and South America, but then these two very different areas being used by the different populations. So we used Bhattacharya's affinity to look at how much overlap there was between the distributions. And we did this firstly by looking at the samples that we took from each colony, so the Prion Island birds and the Bird Island birds, but also to see if this was, these signified different areas at sea. We used a randomization process where we used exactly the same trip data, but we just randomly assigned the colony that it came from. And the assumption is if the population at sea distributions are similar, then by randomly assigning the colony, we're going to get a similar level of overlap. Whereas if they're different distributions, then we'll see a different overlap. And the p-value was determined by the proportion of the randomized trips that were smaller than the sample trips. So what we can conclude from this is that there's no difference in the core foraging areas of the two populations, but there is a significant difference in the home range. So the first thing we've only vaguely looked into is whether this is down to the wind conditions when leaving the colony. And so these plots show the direction that the wind's coming from. And the panels on the left-hand side show a point just off the colony at the point that the bird departs the colony. And the plots on the right-hand show the point 1 hour into the foraging trip and the wind conditions at this time. But I didn't really look into it further, or I haven't really got round to it yet, but I'm not convinced. I mean, the wind conditions seem to be coming from very similar directions at both colonies, although there may be something to look at in the strength of the wind, as we know that they tend to use headwinds or tailed winds depending on wind strength. But I'm still not convinced that this will find anything explaining what's driving this segregation using the wind. So I used some hidden Markov models to separate ARS behaviour. As I've said, I went down to 1 hour resolution, so as our talk the other day stated, this isn't great for the hidden Markov models, but I have been able to compare it with some of the landings data from Bird Island, and actually we were identifying the main areas where ARS behaviour was being seen, although we did miss some of the opportunistic dives on the transiting part of the journey. But I used general additive models to look at some of the key common environmental covariates associated with habitat characteristics, and I modelled both colonies completely separately, yet the same four habitat characteristics came out for both colonies, and looking at the response curves, they're very similar between the two colonies as well. They like shallowish shelf areas in temperatures under 20 degrees, fairly close to the colony, and with increasing eddy kinetic energy, which represents kind of fronts and current speeds. So it looks like they're foraging in very similar habitat types. I don't think there's any difference there driving their distributions. So when we created the models, I used real-time data, so the value of the environmental covariate is that value on the day that the bird was in that cell. So I took an overall seasonal average between January and March and then used the models to project back into the foraging, into the home range area. And we can see that it would predict that birds from both colonies would use these areas to the east and to the west and down to the Antarctic Peninsula, just confirming really that it's not that they're foraging in different habitat types, the birds from different colonies. So moving on from why their distribution's different, because I can't answer that at the moment, so if anyone's got any insight, I'd love to have a chat later. But looking at the consequences of this, we can see the plot to the left just shows the proportion of the population in each of these grid cells. And we can see that from Bird Island, there are relatively few of these darker cells, which suggests that the populations are more highly dense in these small areas of blue cells, than the birds from Frion Islands, where there are a lot more of these blue darker cells, which suggests that the population's a bit more spread out. So the plot in the middle shows all the fishing activity from the Global Fishing Watch that occurred between January and March this year. And we can see that it's really highly concentrated up against the Patagonian shelf. And finally, looking at the overlap from the birds from each colony. And we can see the areas where they're overlapping is really similar. But if you look at the scale, then the overlap index from birds from Bird Island is considerably higher than that overlap index from birds from Frion Island. So just breaking this down into different gear types, we can see that if you look at all fishing together, there's, again, Bird Islands in green, Frion Islands in blue, and so the overlap index from birds breeding at Bird Island is considerably higher for actually all of these fishing types than the overlap index from birds breeding at Frion Island. Although we know they interact strongly with the long lines, there doesn't seem to be so much interaction with the squid chiggers and the trawlers, but just to highlight the overlap is potentially there with all of these different foraging fishing types. So from what we can conclude so far, there are significant differences in the home range between the two populations, but it's unclear what's causing this population segregation. Birds breeding at Bird Island have higher overlap with all types of fishery and thus are more threatened from overlap with fisheries. And this may explain the faster rate of population decline from birds breeding at Bird Island than those breeding at Frion Island. So I threw in some penguins for good measure, and if anyone's got any questions, then I'll be happy to try to answer them. Thanks very much.
Thanks very much for another interesting talk this morning. Are there any questions from the audience here?
Everyone's thinking about coffee.
I think they are, so I might just delay them a little bit by asking a question myself. I was interested by the fact that your Frion Island population have less overlap with fisheries, and therefore the population decline rate was lower there. What do you think this has in terms of implications for the wider population of wandering albatrosses? You know, are we identifying the wrong target areas for conservation, or can we kind of predict which areas need to be better managed based on that overlap?
So we're partners with BirdLife International, who work closely with the fisheries on the ground. So the reason we wanted to do this was so that we can identify where we should be looking at this overlap. But actually it's quite convenient that all the birds tracked have been from Bird Island, and they are the ones more at risk, because it does suggest that we have been targeting the conservation measures at the right areas already. And the areas that the Frion Island birds are overlapping are the same areas as the Bird Islands, it's just a lower amount. So actually, I mean, from colonies elsewhere, I'm not really sure, but from these two colonies, it seems like we're doing the right thing, actually, with our conservation measures that we're trying to really focus work with the fisheries operating off the Patagonian shelf.
And more controversially then, do you think we might be overstating the I guess, the impact on wandering albatross populations from fisheries by actually just focusing on a colony that has the most impact?
I guess you could look at it that way. I think there's been quite a lot of population modelling that suggests that it is breeding adults that are, or not necessarily breeding adults, sorry, the adult survival is the main driver of these huge population declines that we saw someone showed on a graph the other day, sorry, I can't remember who, but we could see the real steep rate of decline. And there's no, if it's not fisheries, it's hard to know what is causing the population declines for the adult birds. So I can see where you're coming from, but I think it's still a good thing to be focusing on the fisheries.
Definitely. Just thought I'd throw it out there. There's a question right at the back corner of the...
Thank you for a lovely talk. I wondered whether there's differences in the population size between the two colonies and whether you would have the data to be able to look at whether sexual segregation is greater at larger colonies.
So firstly, sexual segregation is something I meant to bring up. We've got the feathers to do the sexing of the birds, but we haven't done it yet, so I don't know at all how this is related to sex. The population size is hugely different as well. On Prahran Island, there are about 34 breeding pairs, whereas on Bird Island, there are hundreds and hundreds, I can't remember the exact amount, but 600 or 700, I think, breeding pairs. So the population sizes are different, though it doesn't seem, I know in some seabirds, smaller populations tend to remain closer to the colonies than in others, whereas it looks pretty much like they're both going huge distances. It just seems to be this opposite direction, and I'm not sure quite how population size would drive that. I guess unless the Bird Island birds, the Prahran Island birds aren't going there because of the higher populations from the Bird Island birds, but I'm not really sure, to be honest.
Perfect, thanks. I don't see any other hands up. Oh, hang on. This side. I'll run.
Can you run around? Great talk, thank you. I was just interested in when you were mapping where the birds had gone with the fishing activity, like what did you include as spatial overlap? Was it that there was a tagged bird within like 5-10 kilometres of the fishing activity, or was it within a day period, or what was your brackets for counting it as an overlap?
Yes, I didn't explain that very well. I downloaded all of the fishing data that's happened between January and March in each grid cell, which I think is about 10 kilometres. So it's the hours of fishing, total hours of fishing between January and March in the 10-kilometre cell, and then so we calculated the proportion of the birds in that cell, and that's how we looked at the overlap, by multiplying one by the other, I think. Yeah.
Perfect. Well, I think it's time for caffeine. I'd just like to thank all the speakers from this morning's session for actually keeping us awake.